been talking about the cell and all the parts of the cell. Now we're going to really look into how cells get things in and out of itself. And this is very important because of a concept called homeostasis. Okay, homeo means stable. So organisms need to be able to maintain a balance, a stable internal environment. So I always use the example of when you have a fever, you no longer have a stable internal environment in your body because the temperature has increased, and it's usually due to your body fighting off something. The temperature increases in order to kill that substance, and then also to trigger your immune system to come in and to help fight that off. So your body and cells on an individual level have mechanisms to try to keep conditions stable. So when you think of homeostasis, think of stable internal conditions. I mentioned temperature, but we also have pH. We have buffers that we've talked about in our body that keeps our pH levels where they need to be. Blood has a very narrow pH range that is slightly basic, and it has to stay within that range for enzymes to work properly. Concentration of different materials has to be the same, and so forth. And the cell membrane is what we're going to look at first, is what is going to be controlling this, especially things that come in and out. This is a picture of the cell membrane that you will see a lot. Notice that it has a lot of different parts. These blue, oval, or odd-shaped things here, these are your proteins that are embedded within the membrane. You have, this is a special type of protein here that is is a way of communicating. You have in here cholesterol that helps provide support. You have glycoproteins. These are proteins that have little sugar molecules that are attached to it. You could have carbohydrate tails just floating about. And all of these with the circle and the little tails here, these are phospholipids. And it's called a bilayer because you have two. The tails are hydrophobic, and the heads, which are the circles, are hydrophilic. And so things are not going to come through here very easily. And so we say that the cell membrane is selectively permeable because it kind of chooses. Another term you'll see is semi-permeable. So it just doesn't let everything come across. It is selective. It kind of chooses. It works as a gate. And when we're talking about transport, we're talking about two different things. You're going to hear the term passive and active. So think about passive being you're just kind of sitting around, you're not really doing a lot. Active, think about when you go to PE, you're playing basketball, there's energy you're using. So passive is what we're going to look at today. This is no extra energy. The cell does not have to suspend or use any energy for any of this. All right, we've got something called high concentration to low concentration. So think about if you go to a concert and it's crowded, you're in a high concentrated place. All right, if you went to a concert now, due to COVID, you're going to be in a lower concentration because they're not going to let as many people in. So high concentration, everything is kind of squished together, to a low concentration where they're spread out. And we call that moving down a gradient, down the concentration gradient. Things will naturally go that way. If I were to spray some perfume or cologne on this end of the room, eventually Austin over there will smell it. It will move. It's very concentrated here, and eventually the smell would get over there. That's kind of moving down the concentration gradient. Active transport, which we'll talk about tomorrow, is sort of extra, you need energy. And we're going to learn about this energy. It's called ATP. That'll be a part of our next unit. And this is what is going to be used in order for cells to either bring in large molecules or to expel, send out large molecules. And it's going against the gradient. So it's going from a low concentration to a high concentration. So these are going to be important things to try to associate with what we're doing. So to give some examples, there are three types of passive transportation. We'll probably deal with diffusion and facilitated diffusion today and get into osmosis and then the active transports tomorrow. And active transport, you've got molecular pumps, exocytosis, and endocytosis. Some other terms to review from physical science. Solute, this is what's going to be dissolved. Solvent is what's doing the dissolvent. Normally that's going to be your water. These solute are going to be larger particles that's in the water. And then the solution is a uniform mixture of these. Concentration is just the amount of solute that is dissolved in a solvent. So just to be familiar with those terms. So to specifically look at passive transport, we have just simple diffusion. This is just like I did with the example when I said if I spray cologne and it spreads out, that's simple diffusion. It is just going to go from a very high concentration to a very low concentration. And examples are going to be small molecules like oxygen, carbon dioxide. So there's a lot on this side. So they're going to move through this dashed line, which is the cell membrane, until it's balanced out, until it hits equilibrium. So that's simple diffusion. These are things that can get through the cell membrane. Now, facilitated diffusion is still diffusion, but there's going to be a protein here that will open up, or that is open, and that will allow only certain larger molecules to go through, like glucose, which is sugar, or ions like sodium, which is in salt. And so they're still going from a lot to an area where there's little, but they're having to go through a transport protein. So it's being helped, it's being facilitated, but still no energy is being used. And so we're going to kind of stop right there, and I'm going to pick up with osmosis tomorrow.